Is it audible? Okay, it is. Okay. So in biotechnology, we completed this biotechnology application, that is insulin production. That is the adult diabetes, it is managed by using what it is the insulin. And insulin, the conventional production is by slaughtering or killing the pigs as well as the cattle, and we are getting from what it is the pancreas. But that causes some unwanted immunological responses. A novel strategy we adopted is what is responsible for the production of the insulin. The same is cloned into what it is the plasmid of what it is equally. So two chains which are made, A and B chains which are made, which was introduced into what it is equally, and the two polypeptides which are combined by means of disulfide bridges, and the insulin is also produced initially as what it is proinsulin, having A, B and C chains, that is 20, 30, 33 amino acids, 21, 30, 33 amino acids, and later during processing which polypeptide is removed, the C polypeptide is removed in Golgi complex. So A and B chains which are linked. In case of the first chain, A chain 7th amino acid, and in case of the B chain it is the 7th amino acid, that is cysteine or methionine, that is having what it is sulfur, and 20th amino acid in case of the A chain, 19th amino acid in case of the B chain, disulfide linkages. Okay. And Eli Lilly, an American company, the first time produced what it is the human insulin, what do you call humulin, and recently the Eli Lilly and Ranbaxy company collaborated. With their collaboration, absorption is so quick, the production, what do you call it is Humalog, which absorption is quick than what it is earlier insulin production. To that extent, we completed in case of biotechnology applications. And in the diagnostic methods, effective treatment it depends upon early diagnosis and understanding pathophysiology. It is very, very important. So diagnosis is important. At the same time, understanding the pathophysiology is very, very important. So the physiology of the host due to attack by the pathogen, such a study is also important. All contributed in case of what it is effective treatment. You diagnose it, but we don't have the idea. So there is no understanding ability of observing pathophysiology, there is no use. So early diagnosis, understanding pathophysiology are equally contributing for what it is effective treatment. And that's why for pathophysiology, many NEET aspirants wants to make entry into the government colleges. Why? Because they are exposed to number of what it is the patients, they can study well than what it is the private colleges. So getting the seat in case of the government college, in case of what it is as far as the NEET concerned, because they're exposed to number of the patients, that is very, very important. Understanding of the pathophysiology is very, very important. And diagnosis is also what it is important, all contributing for what it is effective treatment. So we must have the clinical knowledge than what it is theoretical knowledge. Theoretical knowledge is important. At the same time, clinical knowledge is important. How we are going to get such a clinical knowledge? When we expose it with what it is the more number of patient. The more number of patient possible only in case of the government hospitals with the different diseases. So it is possible to know about what it is pathophysiology. Understanding of what it is altered physiology of what it is the host. That is altered physiology of host due to pathogen. So symptomatic study is very, very important. Okay. So conventional diagnosis. The conventional diagnosis, that is serum and urine analysis. With the conventional diagnosis, symptoms manifested and identified. Undoubtedly, symptoms manifested, identified. But by that time, the pathogen concentration very high, the bacteria. By that time, pathogen concentration high, virus or bacteria. So conventional methods having some constraint. So as earlier we said, for effective treatment, early diagnosis, it is very, very important. But by the time the concentration of pathogen already it is high, symptoms are manifested. That is the constraint in conventional diagnosis. So we're going to method, the modern method like polymerase chain reaction, ELISA, and RDN technology, that is non-conventional methods we are opting. And modern non-conventional methods. The modern non-conventional methods. That is PCR, polymerase chain reaction, and ELISA, enzyme linked immunosorbent assay, and RDNA technology, the recombinant DNA technology. In case of the non-conventional method, here symptoms are not manifested. the pathogen concentration very low. So testing is the method. Okay. Symptoms are not manifested. At the time pathogen concentration it is very low. So the pathogen nucleic acid amplified to several billion copies, that is possible by means of what it is polymerase chain reaction. So pathogen concentration is very low. So how can you amplify this nucleic acid by means of polymerase chain reaction? The polymerase chain reaction is a routine method in HIV suspected, that is AIDS patient. The mutations in case of what it is cancer suspected patients, the PCR, it is a routine method. So polymerase chain reaction. So symptoms are not manifested, but pathogen concentration it is very low. So by amplifying what it is the genetic material nucleic acid, we can detect. In conventional method, symptoms are manifested. By the time the concentration of the pathogen already it is high. And this PCR, the routine method, HIV suspected AIDS patients, and cancer suspected patients, observing what it is the mutations, and many other diseases, it is the PCR, it is the method. And ELISA, that is enzyme linked immunosorbent assay. So you have the detailed account in case of what it is the zoology part. Till then it is ELISA enzyme linked immunosorbent assay. It is based on antigen antibody interaction. In host cell due to pathogen antigens detection. Infection by pathogen produce what it is antigens in host, antigen detection. These antigens which are produced by the pathogen, and to combat this antibodies. So detection of the pathogen by the production of the antigens or production of the antibodies against what it is antigens. What do you call the test? It is ELISA, enzyme linked immunosorbent assay. And other it is RDN technology. By using cDNA, that is complementary DNA. And this is radio labeled what it is ssDNA. You should identify what it is complementary DNA. So it is not detecting what it is the complementary DNA. it got mutated. It got what it is, the mutated. So it cannot detect. In case of, so auto radiography using what it is, X-ray film. So detecting what it is the complementary gene by using what it is the cDNA in case of RDNA technology. We are not going to detect what it is the particular gene because the particular gene already got what it is mutated. So it is not observed in case of X-ray film. So these are the modern techniques, the quick diagnostic methods for what it is effective treatment. So RDNA technology, ELISA, PCR are the quick diagnostic methods, the non-conventional methods. So symptoms are not manifested, but pathogen concentration very low and the amplification of the nucleic acid to several billion copies. So radio-labeled ssDNA is nothing but what it is, a probe. It is going to identify the clone or colony. And it is not going to be hybridized because the gene already got what it is mutated. So mutations happen in case of the particular organism. So those are biotechnological varieties, the applications. So insulin production, other biochemicals, vaccines, and that is diagnostic methods. And biotechnology having applications in case of what it is the plants. Earlier we said what it is transgenesis and transgenic plants. Transgenesis, transgenic plants. Transgenesis, modifying genetic trait of what it is organism, the manipulation of the gene, the tailoring of the genes as you decide, transgenesis. A transgenesis adopted in case of the plants what do you call it is transgenic plants. Suppose bacteria, fungi, the plant. So desirable genes are transferred from bacteria or fungi to the plant. Desirable gene transferred from the bacteria, fungi to the plant. What do you call transgenesis? And plant it is transgenic plant. Genetically GMO, genetically modified organism or GM crop. And these transgenic plants are used as a bioreactor for production of medicines, chemicals and antibodies. It is a type of or it is the forming. Such area of the forming, what do you call it is? It is molecular forming. So here transgenic plant act as bioreactor. In earlier said that bioreactor is different. Biotechnology, that bioreactor is a stainless steel vessels in which raw materials are biologically converted into what it is the products. Here bioreactor it itself what it is the plant. Okay. So the transgenic plants act as the bioreactors for production of chemicals, medicines, antibodies called molecular forming. It is a kind of forming. What do you call it is? Molecular farming. In molecular farming, which are the bioreactor? Transgenic plants are bioreactor. How it made transgenic? By transfer of the genes from unrelated organisms like bacteria and fungi to the plant. What do you call? Transgenesis. The plant it is transgenic plant. Farming it is called molecular farming. So in these transgenic plants, in case of for varieties, vitamin A. So for production of golden rice, type A variety of varieties rice into this genes from daffodil plant and Erwinia bacteria. Daffodil plant from Erwinia bacteria, and it forms precursor for vitamin A, beta carotene that forms vitamin A. It once appeared in the NEET 2019. So daffodil plant and Erwinia bacterium, from these the two genes which are transferred to type A, a variety of the rice, and for vitamin A production, what do you call it is? Golden rice. Because of the carotene, orange, yellow colored can be seen. What do you call it is? The golden rice. Because it has to be imported in case of the grain, but not in case of the leaf. Leaf having carotenoid pigment, but the grain it is not having carotenoid pigment. So it is imported in case of it is the grain. So for vitamin A. So night blindness prevented in this golden rice, that is vitamin A. Type A, a variety of rice, instrumental rice variety into which the genes from these organisms are introduced. So it is golden rice. For any type of MCQ, so which is having in golden rice vitamin A. Precursor for vitamin A, beta carotene. Beta carotene produced by means of type A variety of rice after introduction of the genes from daffodil plant and Erwinia bacteria. The first one, golden rice. And second, basmati rice, that is flavored rice, abiotic and biotic stresses. Flavored rice, abiotic and biotic stresses. For particular character with basmati rice. Herbicide tolerant varieties. In this, soya bean or Roundup Ready. It is a trade name, Roundup Ready soya bean. So soya beans which are unaffected when used for herbicides to eliminate varieties, the weeds. Soya bean it is unaffected. Herbicide usage to eliminate herb weeds. So Roundup Ready soybean, it is herbicide tolerant variety. That is the second one. And tomato, it is flavor saver transgenic tomato, that is bruise resistant and delayed ripening, more shelf life. So post harvest losses reduced. Post harvest losses reduced because it is having more shelf life, delayed ripening, bruise resistant, and this flavor saver developed by antigens technology. As you earlier said, in case of pest-resistant plant nematode, that is Myriadogon incognitia, RNAi, RNA interference, RNA quelling, silencing, so sense strand and anti-strand both are complementary, prevent varieties translation. The technology it is antigens technology. In case of third one, efficiency in utilization of minerals. So this is the one side of the coin. When you introduce genetically modified organisms into this natural environment, there are some unpredictable changes we cannot anticipate because we introduce the foreign gene into this organism. How it is going to have the impact, the long-term impact, we don't know. It is having its limitations also. So I have to consider certain ethical values and biosafety issues. I have to know about certain biosafety issues. When transgenic organisms introduced into the environment, it is having some unpredictable changes. Transgenic organisms introduced into environment, some unpredictable changes. We cannot predict what are the changes, how it is going to influence, we don't know. In this, direct transfer of varieties, allergens, because the genes transferred from the bacteria, fungi to the plant, we may consume it. So, transfer of allergens that may happen. And gene pollution due to outcrossing. Gene pollution due to what it is? Outcrossing. So when this pollinated, so it may result in what is the gene pollution. And fundamental nature vegetables change. If you take the tomato, there are many modifications in case of the tomato from the conventional to what it is non-conventional. We are producing many tomato having more shelf life because of this technology. So fundamental nature of it is vegetables, it is the changed, and natural evolutionary pattern is going to be affected because we altered the gene. Mutations are slow. That is required in case of evolution point of view, but there is a quick mutations in this. So natural evolution pattern is going to be affected. Natural pattern going to be affected. Okay. Great impact on what it is the biodiversity. So before introducing the genetically modified organism for public use, the product, it should be thoroughly checked. Some validity it is required by authentic organization. Okay. So GAC, Genetic Engineering Approval Committee. The Genetic Engineering Approval Committee. First, it validates GM research. The first one. So it is authentic agency. So any product should get certification from this agency before being it is implemented for what it is the public use. So genetics for public use has to be certified by GAC. So what is GAC? Genetic Engineering Approval Committee. Before any product that is going to be released as a genetic modification, an approval before it is released for what it is the public use. An approval by Genetic Engineering Approval Committee. That is validate GM research. Genetic products for public use has to be certified, that is approved by Genetic Engineering Approval Committee. So some ethical issues, biosafety issues are there. Even though there is a tremendous development in case of the biotechnology, there are many apprehensions, doubts regarding the biosafety issues. How it is going to have the impact in the near future, whether it's going to have the effect in case of any system in our body. So there are many apprehensions regarding this biosafety issues. That is also one key point. So biotechnology has a tremendous improvement, but the products release it is not that much of this what it is the speed because of what it is apprehension, doubts in public with regard to what it is biosafety issues. So usage of biotechnological products not that much than the progress made in biotechnology. Usage of biotechnological product not that much than the progress made in case of the biotechnology, because apprehensions, doubts regarding biosafety issues in public. Suppose if you take the example corn, you are getting it very cheap, the 10 or 15 rupees, the corn. You are getting in the form of the corn flakes. But in Western countries, the corn just banned because of detection of authorities, some impact on the life in near future, not it is instantaneously. Like it is in some countries, the MFC chicken, that is, what do you call it is, the fried chicken, MFC or some other, the hens used. It is also a biotechnological product, genetically modified product. It is also banned in certain parts of the world. So, certain biotechnological product not approved in certain countries, but it was got approved in some other countries like developing countries. Developing countries are poor financially, but we are having much biodiversity. Financially, but poor in case of what it is biodiversity. And rich countries do not approve such methods very quickly than what it is the developing countries. So we will discuss in case of what it is those things in case of ethical values. So usage of biotechnological products not that much than the progress made in biotechnology. Why? Because apprehensions, doubts with regard to what it is biosafety issues. How it is going to have the impact on our life, we don't know. The BT brinjal, it was banned in case of what it is India. So now BT cotton you are using, you are not, because you are not going to consume what it is the cotton. So BT cotton it was not banned, BT brinjal it was banned in India. Okay, up to this. Okay. Okay. And we left in this part only some ethical issues in case of biotechnology applications. So after completion of this part, we're going to have this physiology part. It's very lengthy part, having many lessons in case of the physiology part. Their weightage is much in case of the NEET. As we complete very important topics like molecular biology, biotechnology application, and we're going to start with what it is the plant physiology also. So we left with what it is the 12 or 13 minutes. We'll discuss some of the things with regard to the MCQs in case of the lac operon and the biotechnology applications. Operator plus repressor. Genes switched on. Lac operon is continuous till glucose levels are low. True or false? Lac operon is inducible system. Real inducer of lac operon. Lactose, galactose, allolactose, beta galactosidase. Question number 1, 2, 3, 4. Question number 1, 2, 3, 4. Operator plus repressor gene switched off on. Question number 4. Allolactose. Vamshidhar Reddy, okay, allolactose. Roja, first one, it is operator repressor genes switched on, it is off. False, okay. And question number 2. Lac operon is continuous till glucose levels are low. Yes. Lac operon is inducible system. Yes. Inducer. Lactose. Lactose. Fourth question, real inducer of lac operon. It is allolactose, isomeric form of the lactose, it is allolactose. When lactose, allolactose are given, the best option it is allolactose. If allolactose is not mentioned in case of the options, we have to take what it is, lactose. 4C, right? That is allolactose. Okay. Real inducer of lac operon, allolactose. Dash is ratio of cytosolic and membrane bound enzymes involved in lactose catabolism. If Y gene mutated dash enzyme formed. 5, 6. Question number 5. Question number 6. Dash is the ratio of cytosolic and membrane bound enzymes involved in lactose catabolism. So Palkavit, Roja, 2:1. Yes. 2:1. Permease is a membrane bound enzyme. Remaining enzyme beta galactosidase and transacetylase present in case of cytosol. 2:1. If Y gene mutated, nonsense mutation, dash enzyme formed. The sixth question. Sixth question. No enzyme formed. No. ZYA. In Y gene mutation, Z gene it forms what it is? Beta galactosidase. Beta galactosidase. Okay. For sixth question, beta galactosidase. Okay. Because mutation in Y gene, but not in case of Z gene. If Z gene having mutated, nonsense, there is no beta galactosidase, there is no, or it is permease, there is no transacetylase. Okay. Lacoperon is a polycistronic structural gene. False. 8th. Dash is a ratio of start and stop codon in Lacoperon. 7th and 8th. Question number 7. Question number 8. Lacoperon is a polycistronic structural gene. Dash is the ratio of start and stop codon in Lacoperon. Roja Seventh question, yes, lagoperon is a polycystonic structural gene, right? Because ZYA. Roja, eighth one, 1:1. Okay. 3, 3. 1:1. Polycystonic structural gene, that is 1:1. Seventh question, it is the true. Polycystonic, it is having ZYA gene. More than it is one gene, polycystonic structural gene. Okay. Okay, it is. Okay.